يلا who can read. اجا قاسم. اوكي يلا شباب، عبد الله، قاسم who can read. طيب يلا يا مريم start if you can.
Yes, Zaria.
اوكي مريم او جنى no problem.
يعني بلاش أنا أو هي.
تفضلي يا جنى يلا. خلي مريم بس for serious cases.
Okay.
يلا.
The meningitic leukemia virus CML is slightly pathogenic and infects nervous cells in the framework of studying the immune response against the infection by this virus. 2 experiments were performed.
تمام.
Experiment
1.
Yes, different viruses are injected into mice of different strains Y and Z. The experimental conditions as well as the obtained results are shown in document 1.
فإذا we inject CML into a mouse Y. After 7 days we remove the spleen and then we extract T8 cells. طب we add
Yes.
T8 cells now to 4 media. In the first one, addition of cells Y infected by CML مع T8, we have lysis of these cells. أما addition of cells Y non-infected, no lysis. Cell Z infected by CML, no lysis. Addition of cells Y infected by HIV, كمان no lysis. فإذا the lysis occur only in first medium where you have cells from Y infected by CML plus T8 activated in the same mouse, okay, injected by the same virus. طيب يا جنى, name the specific immune response triggered against a virus and that triggered against a bacterium.
Says against the virus هي cell mediated وبكتيريا هي humoral.
اوكي فاذا against virus هي المفروض
cellular.
cell mediated او cellular mediated، بينما البكتيريا اوكي اللي هي مفروض humoral mediated specific immune response. اوكي طيب تمام ممتاز. continue يا جنى لكن.
Interpret the results of experiment 1.
Interpret the results of experiment 1. Okay? يعني now أنا I need to do the analysis, then I do the okay the significance. طيب. Okay يلا يا جانا start with the first one.
Okay, in the following experiment, the mouse Y was injected by CML virus, and after 7 days, they removed its spleen and extract its T8 cells.
Okay.
And observed 4 results.
طيب.
In first result, we observe a lysis due to the addition of cell Y infected by CML. However, in second result, we observe no lysis when we add cells of Y non-infected. While in third result, we didn't observe a lysis when there was addition of cells Z infected by CML.
Okay.
Also, in the fourth results, we didn't observe lysis when there was addition of cells Y infected by HIV.
طيب.
This shows that T8 activates to specific type of virus that is injected to the body of the mouse to specific type of cells.
طيب Jana, when you say we observe, this is describe مش analyze.
ممنوع استخدم كلمة
ما فيني اكتب observe بالanalyze.
اوكي.
اوكي يعني بالanalyze بتقولي لي مثلا there is lysis in the first medium where we have or where we add cells from Y infected by CML and T8 extracted from a spleen of the mouse injected by CML. تمام؟
Yes.
while there is no lysis if the cells from Y are non-infected, or the cells are from Z but infected with CML, or from Y infected by HIV. This indicates that, okay, T cells activated in a body destroy only cells from the same body infected with the same virus. You see?
Yes.
Okay. طب, let us read it together. Okay, and then if there is anything not clear we can explain it. طيب. فإذن هيدا exercise 28. Okay, يلا يا جنى. Read part 2. Cells Y.
Okay, cells Y infected by CML undergo lysis by T8 cells, which are taken from mice of the same strain Y injected by CML. Whereas cells of the same strain Y that are not infected don't undergo lysis. This shows that T8 cells destroy only infected cells.
تمام.
On the other hand, T8 cells, which are taken from mice of strain Y injected by CML lyse the cells of same strain that are infected by CML, but they don't lyse cells of a different strain Z infected by the same virus CML.
Okay.
This implies that T8 cells lyse only infected cells that belongs to the same strain.
To the same strain. طيب.
بس عندنا كذا يعني كذا يعني significance كذا واحدة.
ما في مشكلة. You can separate them. ما بيأثر.
Okay. Cells Y infected by CML undergo lysis by T8 cells, which are taken from mice of the same strain Y injected by CML. On the contrary, cells of same strain Y, which are infected by another virus, HIV, are not lysed. This implies that T8 cells destroy only the cells infected by the same virus that activated them.
Okay, only the cells infected by the same virus that activated them. Okay, جيد. Okay, تمام. Let's continue to experiment 2. T4 cells now. يلا يا جنى.
T4 cells are cultured in the presence of macrophages and CML. The experimental conditions as well as the results are shown in document 2.
طيب, culture of T4 in the presence of macrophage and CML 2-4 days. بعدين removal of the supernatant of the culture, يعني liquid part only. يعني ما فيها لا T4 ولا CML ولا macrophage. Addition of the supernatant. La culture of activated T8 cells by CML. Okay. Here, the medium where you add the supernatant, there is cellular proliferation, يعني cells develop and divide. While if you don't add the liquid, no cellular proliferation. طيب, determine the role and the mode of action of T4 cells as revealed in experiment 2. When they say, Jana, determine, we do analyze, okay? Then we do significance. We do analyze and then conclusion directly. يعني هي analyze مع conclude.
Okay.
طيب يلا اعملي لي analyze هون لنرجع نطلع conclusion.
Having the culture of T8 cells in the presence of macrophage and CML from 2 to 4 days.
Okay.
We removed the supernatant of the culture and added to.
جانا بس تقولي لي we remove يعني عم تعملي description.
نسيت.
ايه ما تجيبي لي سيرة الوي ابدا. بل شيلي بالريزلت بعدين احكي لي conditions. تمام؟
Okay, نقول in the first result انه there was.
دغري مثلا قولي لي there is cellular proliferation in the medium containing culture of activated T8 cells by CML when we add or when adding supernatant taken from a culture containing T4 macrophage and CML virus. But there is no cellular proliferation for the T8 activated T8 cells without the supernatant. اوكي يعني انا حكيت عن الريزلتس بعدين ذكرت الكونديشنز.
اوكي
يعني بلشت ليكي منين بلشت؟ There is cellular proliferation of culture of activated T8 cells after adding supernatant and removing supernatant culture or supernatant taken from where? From a medium containing T4 macrophages and CML virus. Okay? But these cells do not proliferate without adding this. Thus, as a conclusion, T4, activated T4, stimulate multiplication of T8 recognizing the same antigen. How? By producing a chemical substance. احنا اخذنا بس liquid part. By producing a chemical substance لانه we take only the liquid part. طب, what is the role of macrophage in the culture of T4 cells in experiment 2? What's the role of macrophage here?
تعمل engulfment to express للفيروس.
أول شيء حتعمل engulfment, digesting, and presenting an antigen on its MHC2. صح؟
Yes.
Where specific T4 can bind. Okay, where specific T4 can bind, secreting interleukin 2 to activate specific T8 lymphocytes. But explain, we mention information that we already studied. Okay? الوحيد الاكسبلين اللي بنحط شيء حافظينه. الباقي كله من الدوكيومنتس اللي قدامنا والجرافس والفيجرز والتيبلز. Okay? بس الاكسبلين from something you already studied. تمام؟
Okay.
يلا, the macrophage phagocytes.
The macrophage phagocytes and digests the CML virus. The obtained peptides get associated to HLA class 2
تمام.
molecules and expressed at the cell surface. The macrophage becomes an antigen-presenting cell APC. The APC affixes to T4 cells having specific receptors to the HLA-CML peptide complex, thus activating the T4 cells, leading to the formation of TH cells that secrete IL-2.
IL-2. They secrete IL-2 to activate the T8 lymphocytes. Okay?
Yes.
طيب. Now document 3-2 electronograph made at 2 successive times target cells infected by CML or activated T8 taken from experiment 2. Draw a scheme showing the molecules involved in recognition taking place between activated T8 and target cells. Meaning the receptors that I will draw in this figure يا جنى. لنقول كيف ال T8 تعرفت على الانفكتد body cell.
T4.
لحظة. عندك T8 وانفكتد body cell، ما دخل T4 هون.
ليش ما هي اللي حتعرفهم؟
لا
إنه هي بتبع
هلا
هي بتعمل
اوريدي صارت
activate؟
ال T8 activated.
اوكي.
بس انا هلا اللي عم بسألك عنه مين الريسبتورز هون involved؟ مين الريسبتورز؟
T8.
T8. شو الريسبتور تبع ال T8؟ شو اسمه؟ receptor of T8, what is it called?
It says CD8.
CD8 برافو. برافو أحسنتي. CD8. بينما receptor of the infected body cell carrying the antigen شو اسمه؟
MHC1.
MHC1. اوكي MHC1. وضحت؟
Yes.
اوكي فاذا انا if I want to make a drawing I have to draw CD8 اوكي مع ال MHC1 carrying peptide from where؟ carrying peptide of the CML. يلا هبعت لكم البيكتشر.
شو بتعمل CD8؟
ال CD8 هي على السيرفيس اوف T8 cell صح؟ ما بدها تتعرف انه هون عندك infected cell لحتى تعمل لها destruction.
اوكي.
بدها تتعرف بدها تعمل double recognition. شو يعني double recognition؟ double recognition يعني بدها تتعرف في نفس الوقت على ال MHC1 وعلى الانتيجين on its surface. يتعرف على الاثنين مع بعض. اوكي نتعرف على الاثنين مع بعض. اوكي
Yes.
طيب اوكي طيب اوكي the last part now explain mechanism. يلا يا جنى صرنا عاملين كذا وحده. كيف الميكانيزم T8 destroy infected body cell؟
It says إنه T8 cells try to enter the target cell.
T8 ما بتفوت على التارجت سيل. لا T8 ما بتفوت.
مش إنه يعني تتعرف عليها أول شيء.
طيب تعرفنا عليها. شو بنعمل بعدين ك T8؟ What does it do؟ The T8
بتطلع secretes interleukin.
انترلوكين ما خاصة بالانترلوكين. ال T8 عندها بس نوعين من الانزيمز ما في غيرهم. شو هن؟ يلا يا مريم what are they؟
البرفورنس هن بيعملوا حفر بالinfected cell والجرانزيمز هن they destroy it و they cause the lysis of the infected cell.
فإذا اوكي البرفورين make holes in the cell membrane of the infected cell بفوت منهم الجرانزيم okay and destroy the DNA of the infected cell. The DNA of the infected cell. Okay. فإذا هول ما في غيرهم. What happened here? Oh, sorry, what a part is not there. يلا يا جنى read the answer please.
The T8 cell performs the double recognition by fixing the HLA-CML peptide complex of the target cell. Time 1. It secretes perforin molecules that form a channel through the plasma membrane of the target cell. That then it releases granzymes that penetrate the target cell through the polyperforin channel, leading to the degradation of its DNA and consequently to its lysis. Time
To
2.
its lysis يعني to its destruction. To its destruction. صح؟
Yes.
Clear؟
بس هولي صراحة ما كنت كتير بعرفهم بس إنه هلا صاروا كتير.
طيب يعني هلا واضحين؟
Yes.
Okay شباب صبايا any question here? Anything you don't understand? Anything you don't understand? طيب فإذا as you can see هلا they are repeating the ideas بس كل مرة بطريقة شكل بس نفس الانفورميشن بالنهاية. اوكي نفس الانفورميشن عم تنعاد لأنه خلص هو same concept. عندي ماكروفيج، عندي T4، عندي T8، عندي B lymphocyte. هل relation between them؟ فمرة بيسألوا عن T4، مرة بيسألوا عن T8، مرة بيسألوا عن B lymphocyte and so on. That's it. ما في شيء جديد حيكون. لو جاب لي نيو فيروس بس أكيد حنتبع نفس الباثواي، نفس الميثود. اوكي نفس الميثود. طيب يلا يا مريم 29 we do 29 together please. ولا someone else wants to read؟
أستاذ أنا عادي بعمله بس إذا في حدا بده.
طيب anyone else wants to read؟ محمد where are you؟ محمد يلا يا مريم read.
A temporary hypertrophy swelling of the lymph nodes is observed in an individual infected by an antigen like the tetanus toxin.
اوكي فاذا بيصير في عندي swelling بالlymph node. اوكي when individual infected by an antigen مثل التيتانوس توكسين. ما بتلاحظوا when you have an infection in your throat التونسلز عندكم بيورموا؟ شو هن التونسلز؟
مزلعين.
بسموه البنات دينين.
Yeah.
هذول بيورموا، ما هن هول احد انواع الليمف نودز. اوكي
Yes. In order to better understand the mechanisms involved in the swelling, the following experiments are performed.
طيب experiment 1.
The constituents of the lymph nodes of this individual are studied by using radioactive markers. Microradiographs are then performed. The radioactive labeled zones appear in black on the microradiographs.
اوكي document 1.
Document 1 shows the results of labeled radioactive B lymphocytes X and the labeled of radioactive T lymphocytes Y.
تمام. اوكي deduce the level or localization of each lymphocyte population at the level of the lymph node. Okay, where you see the B lymphocytes are found, where the T lymphocytes are found.
استاذ البي lymphocytes بالإكس من فوق.
البي lymphocytes شكلهم بالfollicles.
Yes.
والتي lymphocyte interfollicular zone.
Yes.
اوكي طيب فاذا هيك نحن اتفقنا على هيدا الموضوع انه البي document 1 shows only the follicles appear in black after using radioactive marker B lymphocyte. On the contrary, black color appear interfollicular zone in case radioactive labeling of T lymphocyte. This shows that B lymphocytes are localized in the follicle while T lymphocytes are localized at the level of the interfollicular zone. تمام. Okay. Document 2.
Document 2 shows the microphotographs of the cells identified in the lymph node of the individual who is infected with tetanus toxin.
تمام. فإذا طلعنا صور two type of cells للlymph node of individual infected with tetanus toxin.
Yes. Specify the type of immune response triggered against the antigen and revealed in document 2 هو humoral immune response since plasma cells and B lymphocytes are
البلازما سيلز
هن الأكتيف B lymphocytes.
هن active B lymphocyte يعني B lymphocyte transform into plasma cells to secrete antibodies. That's why you called it يا مريم humoral. Okay, humoral mediated specific immune response. طب what is the molecule secreted by this plasma cells?
The antibodies?
أكيد antibodies.
Yes.
And how the plasma cells adapted to the secretion of these molecules?
كيف أصلاً adapted?
يعني what do you see difference in a plasma cell different than B lymphocyte بتساعدها انها تعمل secretion to a large amount of antibodies.
Yes, because it's activated.
ايه بس شو اللي مبين فيها different؟
استاذ
كشكل
انه عندها rough endoplasmic
reticulum. اول شيء عندي large cytoplasm, larger cytoplasm.
Yes.
Containing a large amount of rough endoplasmic reticulum يلي هو specified in secreting شو antibodies proteins.
Yes.
هن proteins بالنهاية. اوكي. فإذا الاميون ريسبونس here اللي قلناه
هو الهيومورال.
هو اتفقنا انه هو الهيومورال. Okay, immune response trigger tetanus is specific humoral immune response since document 2 shows the plasma cells that are antibody secreting cells resulting from differentiation of B cells upon their activation by tetanus toxin. These cells are the effectors of specific humoral immune response. 3-1 anti-tetanus toxin antibodies. Okay. The size of plasma cell is bigger than that of the lymphocyte, 12 micrometer, is higher than 9.3 تقريبا of these cells who have well-developed cytoplasm, rich in rough endoplasmic reticulum, involved in a protein synthesis. And since antibodies are protein, therefore plasma cells is a cell-adapted secretion of antibodies. Okay, very good.
Yes.
Let's look at experiment 2.
Experiment 2. Cells are extracted from the lymph node of a guinea pig, which is injected with tetanus toxin and from the lymph nodes of a healthy guinea pig.
Guinea pig B. They are then purified to obtain
to only obtain B and T lymphocytes. The results are schematized in document 3.
Okay. Interpret the results. What you can see as a difference?
إنه الجنوا pig اللي عنده tetanus toxin has more B and T lymphocytes.
More B and T lymphocyte. The number of B and T lymphocyte increases.
Yes. In the
infected one.
Yes.
Okay. Increases in the infected one.
Yes, whilst they are in the normal.
So document 3 shows that the number of B and T lymphocyte in the lymph node of the guinea pig A infected by tetanus toxin is higher than the number of B and T lymphocytes. Okay, guinea pig had never encountered thus the contact with the antigen. الجينيا pig يا تحسين هو كيف الهامستر بس أكبر. اوكي هذا موجود بس بجزر اسمه غينيا قريبه من اندونيسيا. هيديك المنطقه هي كانت تابعه لاندونيسيا ولكن الدول الغربيه تدخلت مشان تعطيها الاستقلال مشان تصير دوله مستقله مشان يضلهم يصير في مشاكل بينهم وبين اندونيسيا. Thus the contact with the antigen favors the multiplication of B and T lymphocytes. Okay. طيب justify referring to what precedes temporary hypertrophy of the lymph node. Now, can you now explain or justify why lymph nodes they have a hypertrophy يعني swelling.
أستاذ لأنه there was an increase in B and T lymphocytes يعني في immune response ومن الواحد بتاع immune response هو swelling.
اوكي لا لحظة، السويلينج هون مش بسبب كانفلاماتوري ريسبونس، هيديك بالانفلاماتوري يا مريم.
استاذ بعرف بس انه زادوا البي ان تي lymphocytes.
ايه يعني لما يكون في انفكشن B and T lymphocytes are activated. So they start dividing and increasing in number. صح؟
Yes.
Leading to increase in the volume of the lymph node. لما زاد عددهم هالشيء خلى الليمف نود increase in volume. Okay? Yes. Explain the role of T lymphocyte involved in the immune response revealed in document 2. هون قصده يا مريم على أي نوع من التي lymphocyte؟ التي 4 ولا التي 8؟
لا أستاذ هون التي 4.
التي 4 ما دخل التي 8.
Yes. إنه هو بيعمل activate للبي lymphocyte.
Exactly, they activate the B lymphocytes.
Yes.
فإذا عندي the revealed immune response triggered is humoral effector cells are B lymphocytes. Once B lymphocytes are activated by the antigen, they can only proliferate in the presence of interleukin 4 released by T4 lymphocytes that are activated by the same antigen and differentiated into interleukin secreting cells. Thus, T4 lymphocytes are responsible for the proliferation of activated B lymphocytes. Clear?
Yes.
Everyone?
أستاذ
تفضل.
أستاذ فيك تعيد 3-1 3-2 ما فهمتهم.
3-1 البلازما سيلز شو بتعطي يا محمد البلازما سيلز؟ البلازما سيلز هي منا activated B lymphocyte؟
بلى.
شو لازم تعمل تعطيه يعني شو تعمل production of what؟ antibodies. بس هون انت بتقلي انتي لنقول تيتانوس توكسين انتي بودي مشان تكون انت specific.
Yes.
طيب السؤال الثاني how the plasma cells adapted to the secretion يعني كيف التحول ما بين البي ليمفوسايت والبلازما سيل هيدا ساعدها انها تشتغل هالشغل هيدا اللي هو secreting antibodies. اول شيء بتلاحظ يا محمد انه عندها bigger cytoplasm بالسايز كبرت صح؟
صحيح.
اوكي هيدا السيتوبلازم بيحتوي على كمية كبيرة من شو؟ من الرف اندوبلازميك ريتيكولوم يلي هو مسؤول عادة عن production of proteins. والانتي بوديز هن بروتينز. فانت زاد عندك الاندوبلازميك ريتيكولوم كرمال تقدر تفرز كميات اكبر من الانتي بوديز. اوكي؟
اوكي.
وضحت؟ طفي القصم بسرعة.